Today, our next speaker is MBBS from Shandong University and a PhD from University of Tennessee. And he is a postdoc of Dr. Martin Pera from the Southern University, Southern California University of and also a postdoc from Salk Institute of Juan Carlos. Dr. Jun Wu is currently an associate professor in the Department of Molecular Biology at the University of Southwestern Medical Center. Dr. Wu's work has contributed to the development of novel culture systems and methods that enable the generation of new stem cells for basic and translational studies. Dr. Wu has expanded the spectrum of pluripotent states by capturing the mouse embryonic pluripotent stem cells with distinct molecular and phenotypic features from different developmental stages. And some of these culture conditions developed in mice enabled the generations of pluripotent stem cells from many other mammalian species, including human, non-human primates, and ungulates. In addition, Dr. Wu has developed efficient and versatile blastocyst complementation methods for in vivo generation of functional tissues and organs from cultured pluripotent stem cells and several stem cells derived embryo models. Dr. Wu has received several awards, including University of Texas Southwestern Endowed Scholar in 2017, Crypt Scholar 2017, New York Stem Cell Foundation Robertson Stem Cell Investigator Award in 2022, the ISSCR Outstanding Young Investigator Award of 2024. A person with a vast experience in stem cell biology, we have the privilege to listen to him today morning here in India. Ladies and gentlemen, please welcome Dr. Jun Wu from US. Jun, stage
yes.
is yours.
Yeah, all right. Thank you very much, Sojoy, for the generous introduction. And good morning, everyone. This is Jun Wu. I'm here is a little bit late. Hopefully, I can have a clear-minded to give you my presentation in the go base. So let me share my screen real quick. All right, you can see it, right?
Yeah.
All right, so today my talk, do you see my screen or my presenter screen or just the full screen?
Yeah, we can see the full screen.
Full screen, good. Okay, all right. So without further ado, today I'm going to talk about a few projects in the lab, which is based on this very unique cell type called pluripotent stem cells. And pluripotent stem cell, as you probably all know, is very unique cell type. It can be generated from several different sources. One of the important sources from the early embryo, we can take a blastocyst. Let me get a later point. Take a blastocyst and culture the blastocyst in vitro culture condition, giving the right condition, growth factors, and combination of signaling inhibitors or activators. Then we can derive what we call embryonic stem cells from early embryo. And also in 2006, Shin Yamanaka and Takahashi, they pioneered the approach to generate embryonic stem cell-like cells from somatic cells through this reprogramming process by forced expression of 4 Yamanaka factor or 4 transcription factors that are specific to pluripotent stem cells, and then they can stabilize, or we call it induced pluripotent stem cell iPS cells from somatic cells. And once these cells are stabilized in culture, they have these 2 very unique properties. One of them is self-renew. They can making copies of themselves over and over again. And the other is called pluripotency. It is the ability of a single cell to give rise to essentially all the cell types and tissues and organs in our body. So one of the main direction of my lab, when I started my lab in 2017, is to use pluripotent stem cells to generate three-dimensional early embryo-like structures. Throughout the past 6 years, we have been successful in generating several different type of stem cell-derived embryo models, including blastocyst-like structures, or called blastoid, from a mouse, human, and more recently from the bovine culture bovine stem cells. And last year, we published a protocol we can generate a peri-gastrulation human embryo model, which is covering about 2 to 3 weeks of human development using another cell type, extended pluripotent stem cells. And more recently, there's some unpublished study from my lab. We were able to generate a more complete human embryo-like structures containing actual embryonic cell types as well as embryonic cell type. So today, I'm going to talk mostly about human blastoid and human perigastroid. So one of the first questions you may ask is that why to model early human development using stem cell? Why do we need to generate all these different embryonic-like structures? And there are many different reasons, and especially Jenny Rosang wrote a recent very beautiful review, talk about the rationales why human embryo models are important for early human development. But there are several things I want to point out here. Is one of them is we can provide accessible experimentation, meaning that by using pluripotent stem cells or culture stem cells, we don't have to always use embryos, because human embryos are really limited in quantity and very ethically challenging to obtain human embryos at the early developmental period. By developing a model, an in vitro model starting from stem cells, we can reduce the burden of using human embryos for research. And second is because the pluripotent stem cells can be grown in large quantity and essentially in unlimited quantity. And as you can imagine, if we can harness these cells to generate early progenitors, early tissues, we can essentially provide unlimited embryonic tissues, which has potential for regenerative medicine, disease modeling, and many other downstream applications. And third, and I think is one of the most, is that by quoting Richard Feynman's work, famous saying is that what we cannot create, we don't fully understand. So by having this bottom-up approach, by using stem cells to study their crosstalk through the process of self-organization, may help us to understand how cells are interact with each other during early development, so that they can generate this sophisticated, properly organized 3D-dimensional structures that are that give rise to all the tissue and organ later on during development. And this is particularly important because, as you know, during early human development, from weeks 2 to 4 is essentially what we call the black box of human development. We don't really have access to the materials during this gestation period, so it's very challenging. So by having a stem cell embryo models, we can bypass some of the limitations. So in order to generate human embryonic structure, first we need to know what type of human pluripotent stem cells we need to use. Fortunately or unfortunately, there's a lot of different type of human pluripotent stem cells have been reported in the literature in the past several years, and largely they can be grouped into 3 categories. The one type of pluripotent stem cell called naive pluripotent stem cells. They are more similar to this pre-implantation epiblast from the human embryo, such as a embryonic day 6 or 7 epiblast. So these are before implantation. They are naive in the way that they are not lineage restricted. They still have retained the potential theoretically to give rise to all the tissues or organs in our body. And then there's another group of pluripotent stem cell called prime pluripotent stem cells. So these actually are the first human embryonic stem cells are derived in these conditions. In 1998, James Thompson used human blastocysts and cultured them in a culture media containing FGF, and also their secreted activating other factors that stabilize the human embryonic stem cells in actually a post-implantation stage. Now we know that they are corresponding to this peri or pre-gastrulation epiblast stage. In humans, it's about embryonic day 12, 10 to 12. And then there are a group of proponent stem cells that are sitting between a naive and prime proponency. For lack of a better name, we call them intermediate proponent stem cells, or Austin Smith often refers to this formative phase of proponency. So these are having features of sometimes both naive and prime, but also they have their own unique features. For example, in mice, the formative proponency are characterized by a direct response to BMP signaling and give rise to primary germ cell-like cells. But human is more complicated than that because the PGC specification is still unknown and clear at this stage, which tissue they come from and what stage of the development they can be derived from. So first, I'm going to talk about our efforts for the past few years to generate human blastocyst-like structures using cultured stem cells. So the cell type that we use for generate, we call it blastoid, is this naive human proponent stem cells. So the reason why we use this particular cell type is that, including study from us and also several other reported papers in the past, showing that under certain conditions, human naive proponent stem cells can give rise to 3 cell type at least. So a cell type called epiblast-like cells, trophoblast-like cells, or hypoblast-like cells. So these 3 cell types are essentially what you found all 3 foundational cell types in the human blastocyst. So unlike naive mouse proponent stem cells, some of you may know, mouse ES cells are difficult in generating actual embryonic trophoblast tissue, but human proponent stem cells are more capable of generating actual embryonic tissues because they're more plastic than the naive mouse cells. So by this observation, the fact that you can generate all 3 cell types typically found in the human blastocyst, that prompt us to test this a condition, a protocol, develop a protocol to essentially sequential treat naive proponent stem cells with 2 different culture medias. The first media we called trophoblast differentiation media. And so we let some of the cells differentiate into trophoblast. And then we switch the culture conditions and let some undifferentiated cells differentiate into another actual embryonic lineage called the hypoblast. So by the end of day 7 of culture, we ended up with all 3 cell types. This SOX2 positive cells are labeling the epiblast-like cells, and GATA6 positive cells are marking the hypoblast-like cells, and then GATA3 here are used as a marker for trophoblast cells. So we can also switch the order of the treatment. We can use either HDM, the hypoblast differentiation media, first, and then switch to TDM or TSM as trophoblast differentiation media, and we can do the same thing. So the order really doesn't matter in the 2D configuration. And then a couple postdocs came to me and tell me that why not we can grow them in 3D using essentially the same similar culture conditions. Now we, instead of 2D, we switched to 3D, started with 20 to 30 naive human proponent stem cells cultured in this specific culture condition called 5ILA. Is initially developed by Rudolf Janisch at MIT. And then we either treat them first with hypoblast differentiation media, in this media containing FGF, activin A, and chiron, which is the activator of the canonical Wnt pathway. And then we switch to TDM, a trophoblast differentiation media. And at the end of the differentiation, you can see that we can generate these structures are very similar to your human blastocyst, containing a cavity and also the inner cell mass, and then the outer layer of trypanoderm. So again, we can do this the other way around. We can use the same starting 20 to 30 cell aggregate, treating them with trophoblast differentiation media first, and then switch to hypoblast differentiation media. We can do the same thing. We didn't observe any differences. So the efficiency initially was not very high. It's about 10%, 10 to 15%. But last year, we were able to further improve this protocol by simplify the HDM and TDM components, especially the TDM we adding the factor LPA, which is a hippo pathway activator inhibitor, and then we were able to robustly and efficiently generate human process-like structures from across different ES cells and iPS cells with high efficiency. So this is an image showing a typical experiment done in the lab. So we started this using this micro well. So each well is micro contain 1,200 mini wells, and this wells has about 20 to 30 cells to start with. And at the end of the differentiation, you can see that essentially in all the wells, you will see a structure looking like this, very similar to a human blastocyst-like structure, blastocyst containing a cavity in your cell mass. And we perform the immunostaining. Again, here we use SOX2 as an epiblast marker and SOX17 as a hypoblast marker and GATA3 as a trophoblast marker. You can see by comparing to human blastocysts, a human blastoid contain essentially the same similar cell types that you typically find in the pre-implantation human embryos. And we quantify the number of trophoblast and hypoblast and epiblast in the human blastoid, and compared to the human blastocyst, we were able to see that they are really comparable to a blastocyst around 6 days post-fertilization. And although in some cases we do observe there is a little bit more epiblast in the blastoid structures, and there are a little bit less hypoblast, but in general, they are comparable. And we also perform single-cell RNA-seq analysis. So here, just to show you a map of the reference data set using published human embryo from pre-implantation and post-implantation, and also from the human embryo models. So we first generate this reference map. So basically here shows on the right is a pre-implantation human blastocyst cell type, and showing on the left is a post-implantation human embryo cell types. And you can see here in our original protocol, 77% of the blastoid falls, overlap with the pre-implantation human blastocyst. And with the improved protocol, we were able to observe up to 95% or more those cells are aligned with the pre-implantation human blastocyst. So both the fidelity and the efficiency has been improved using our new protocol. But our original protocol also is a good protocol because it captures a majority of the cells from the human blastocyst. And by using a protocol that published by Magdalena Zerniga-Goetz lab from Caltech, now she's at Caltech, so we could culture the human blastoid post-implantation stages. So this is attachment culture, basically let the blastoid sitting on top of the culture dish, and then the center of the blastoid will self-organize and generating structures looking very similar to early post-implantation embryos. For example, in the OCT4 positive cells, you can see there is a pro-amniotic cavity starting to form. These are the precursor for the real amnion cavity that later on in around day 10 to day 12. And then these are very comparable to human blastocysts growing in the same condition. So this is the human blastocysts grow to about 10 days post-fertilization. And then comparably, human blastoid are very similar. But I have to say that the efficiency of generating these type of structures are much less compared to a human embryo. So meaning that there are some a lot of differences between blastocysts and blastoid. And also at the edge of the outgrowth, we were able to detect the hormone HCG-B positive cells in the trophoblast. And we also take the spin media and measure the HCG levels, and they are very comparable to human blastocysts, suggesting that these trophoblast cells are starting to mature and secreting hormones, HCG hormones. So now we have the system. The question is, what can we use them for? So here I'm going to tell you a couple of applications that we already published, but there are many other potential applications that are ongoing in the lab. We are still working on. So first is to use this blastoid formation to study signaling activities that may be involved during early human development. So use human blastoid as a proxy to understand the signaling dependencies of early human development. So During the generation of the blastoid, we observed that there are two different types of structures. Those are the structures that are successfully give rise to this blastocyst-like structure, have a cavity, inner cell mass. And then there are other structures that fail to form a blastoid. Essentially, it's a solid aggregate. It doesn't have a cavity. So we want to compare these two. What are the differences? What made these structures give rise to a cavity? Right? And then first we look at the composition of these two different structures. It seems like both of the structures contain all three blastocyst-like cell type, but the proportion is very different. So in the blastoid, you see majority of the cells are actually GATA3 positive cells are trophoblast cells, and then you have some cells that are epiblast, OCT4 positive, and SOC17 positive. But in the non-blastoid aggregate, you see the trophoblast cell number and proportion greatly reduced, suggesting that maybe the lack of specification or differentiation or maturation of the trophoblast cells may be the reason why they didn't form a successful form of blastocyst cavity. And by using blastoid formation, and also the blastoid and non-blastoid, we examine several different signaling pathways. And first, we call a JAS system. Basically, it's a low input, like a low input Western blot analysis. We look at signaling activation status. These 5 signaling pathways, including AKT, mTOR, MPK, STAT3, and MAP kinase. We observed that compared to non-blastoid aggregates, so human blastoid, 2 of the signaling, AKT, mTOR, are highly activated, significantly more activated compared to the non-blastoid, but they are still lower than the real human blastocysts. So suggesting that these human blastoid are still different from human blastocysts, but nonetheless, they are better in terms of the signaling pathways, better than non-blastoid aggregates. The other 3 signaling pathways, we didn't see any difference. But the MPK pathway, though, through in another experiment, so this is unbiased mass spec experiment, we found that indeed the MPK pathway is more activated in the blastoid compared to the aggregate. So suggesting this pathway is also important for maybe for the blastocyst or blastoid generations. Then we choose several different inhibitors to inhibit each of these AKT, mTOR, and AMPK pathway. We found that with increasing dosage of this BA3727, which is the AMPK pathway, we were able to see reduction in the blastoid cavity formation efficiency. And then by inhibiting AKT pathway, essentially abolish the ability of the naive proponent stem cells to generate these blastoid structures. And then also using rapamycin, which is mTOR inhibitor, we could also significantly reduce the efficiency of generating these structures. And next, we want to know which cell type these pathway inhibitors impact most. So I want to show you the data on the trophoblast-like cells. All I can tell you is the epiblast-like cells and haploblast-like cells are not affected. So these pathway inhibitors exclusively inhibit the proliferation or maturation of the trophoblast-like cells, which explains why if they failed in the presence of these inhibitors, fail to generate the blastocyst-like cavities in the blastoid. And also we took advantage of the blastoid formation to study atypical or PKC isoforms, because in mouse studies, people have identified that the atypical PKC isoforms are important for mouse blastocyst formation. But in humans, this information is lacking. And first, we look at the published data set on the human embryos. We found that at embryonic day 7, we found 4 different PKC isoforms are highly expressed compared to the previous time. So these including a PKC eta, PKC eta and delta and another one zeta. So we were able to find 3 inhibitors that can target each one of these PKC isoforms. So first we saw that, so here PKC eta using as example, they co-localized with tight junction protein 01, suggesting these genes are not only expressed at the RNA level, but expressed at the protein level in the human blastoid or the blastocyst, and they co-localize correctly with a tight junction. And by inhibiting PKC delta, eta, and zeta, we are able to abolish or reduce the efficiency of generating human blastoid. But as a control, using typical or conventional PKC isoform PKC alpha, we were not able to. There is no effect, suggesting indeed in human embryo, or sorry, human blastoid, the atypical and novel PKC isoforms are important for the cavity formation. So this is similar to what people report in the mouse blastocyst. And also by here, just the image, a representative image showing that by inhibiting each of the PKC zeta, eta, and the delta, we were able to not only abolish the cavity formation, but also the tight junction formation is disrupted, as visualized by this Z01 staining. So in the control situation, you will see continuous Z01 staining along the border of the cells, but in the presence of these inhibitors, you see there's discontinuous or sparsely labeled Z01, suggesting the tight junction is not properly formed. And more recently, we were able to establish the co-culture of the human blastoid with endometrial stroma cells. So endometrial stroma cells is one of the cell types that after human embryo implant, the trophoblast cells will interact intimately with these cell types, the stroma cells. So we develop a co-culture by plating either blastoid or the blastocyst directly on top of this monolayer cultured stroma cells. And because the stroma cells are not be able to culture them, we will not be able to culture them long term. So we have to immortalize them in order to generate these cell lines so that we can continuously using the same cell line for different co-cultures. So we found that indeed the human blastoid can be cultured on the stroma cells. And once we grow these blastoid on top of the stroma cells compared to fibronectin coated plate alone, these blastoid grow much better. We see the outgrowth areas much better. And also here, just a comparison of the human blastoid and blastocyst growing on the stroma cells. They look very similar. They both grow very robustly. And we found that in the presence of stroma cells, here we call IESC, is basically immortalized endometrial stroma cells, IESC. So in the presence of the stroma cells, the trophoblast cell number of the human blastocyst as well as the blastoid, so the blastoid here, blastocyst on the right, they're both same, the number of the trophoblast cells is to be more. So suggesting that the stroma cell can promote proliferation of the trophoblast cells. And so this effect seems to be not dependent on the direct contact, because when we use the conditioned media from the stroma cells and directly use the conditioned media to grow the human blastoid, we could achieve the same effect. So suggesting there are some secretive factors present in the stroma cells, secreted by the stroma cells, that have this positive effect on the blastoid or the blastocyst trophoblast cells, right? And then we perform the mass spectrometry and then look at what in those media, and now we have a list of the current candidate that we can help us to further narrow down. So now we're testing each one of them and adding these factors to the cultured human blastoid blastocyst in the without the presence of the stroma cell, to see whether we can reproduce this effect, this proliferation effect of the trophoblast cells using these factors. And the other thing we noticed that by growing a human blastoid on top of the stroma cells is that at the edge of the outgrowth, we see a lot of cells that have multi-nucleus. So this suggests that the sensitization or maturation of the trophoblast starting to occur. And here, just to show that compared to the blastocyst, we saw the same thing. At the edge of the colonies, you see these giant cells has a multiple nucleus. These are the syncytial trophoblast cells. So the differentiation of the trophoblast progenitors into this cell type. And we quantify these effects, and we found that in the presence of stroma cells, so these in the presence of IESC, immortalized in the matrix stromal cells, we see there's a more cell number of the syncytial trophoblast-like cells. Basically, we're counting the number of cells has multiple nucleus. And we found that this effect seems like dependent on the physical contact of the stromal cell with the trophoblast cells, because the conditioned media here didn't reproduce the effect, suggesting direct contact of the trophoblast cells with stromal cells is important for the differentiation of the trophoblast cells into syncytial trophoblast cells. And here to summarize this part, by using a naive human pluripotent stem cells cultured in this specific culture media called 5-ILA, we were able to develop a protocol using a two-step approach to have the cells to differentiate and self-organize into a human blastocyst-like structures. We call it human blastoid. So once we have the human blastoid, now we can, I talk about today, to co-culture with the stromal cells, and also we have recently established the 3D implantation model in vitro to study the implantation of the human blastoid and also the blastocyst in a dish to help us to understand why in some cases women cannot have successful implantation in a condition called repeated implantation failure. What are the mechanisms, and also to help us to screen chemicals or small molecules that can promote implantation in vitro. And also, I talk a little bit about using human blastoid to model early post-implantation human development. But again, this is very inefficient compared to the human embryos. It's much less efficiency. And because human blastoid can be produced in large quantity, we're looking into using this platform to perform large-scale genetic many other things that are clinical relevant. So I'm going to switch gear and talk about the other model that we developed in the lab in the past year. This is an early post-implantation human embryo model called the human perigastroid. So for this, we use a different cell type. We didn't use naive human pluripotent stem cells, although we tried. It didn't work very well in our hand. So we choose to use this called human extended pluripotent stem cells. So the reason we choose to use this cell type is that this cell type can very robustly give rise to two different cell types, the SOX2 positive cells and SOX17 positive cells. So SOX2 positive cells is very similar to the pluripotent cells, epiblast cells, and the SOX17 positive cells are labeling the hypoblast. So, and after a simple differentiation within about 2 to 3 days, we were able to generate these two cell types. No other cell types are generating using these culture conditions. And more surprisingly, we saw that by using this culture condition, we can actually culture these two cell types together for long period. So this is showing that at passage 12, these two cell types can coexist in the culture dish. And then they coexist in a way that their distribution in the dish is not random. So they seem to organize in this very interesting pattern with SOX2 positive cells sitting in the middle and surrounded by SOX17 positive cells. So these observations suggest to us that there's some kind of crosstalk between these two cell types. And interestingly, when we passage these cells, we don't need to effect sorting. We just passage as if they are the same cell line, and they are able to recapitulate all these patterns even after 10 or more passages. So with that observation, one of my postdocs, thinking maybe we can use a 3D culture to recapitulate this interaction. Maybe can give us a better or more robust post-implantation human embryo models, which exactly what he did by using a 4-step approach. Initially, we grow these EPS cells. Here is about 50 to 100, these green cells. A very, in the beginning, is roughly homogeneous population. And then through this first step, lineage commitment, we grow them in a condition that can differentiate some of the cells into SOX17 positive cell, which is the red color here. And then we remove one of the factors called chiron, which is canonical when activated, but also they can, because the stabilizer bicantini also localized to the cell junction by removing the factor, able to help the cell to sorting into different compartment. So this is cell sorting stage of the differentiation. And then after this step, we essentially took out all the component growth factor. There is no growth factors included in the culture, no chemicals, and we just grow them in a permissive media containing serum and the KSR, and give them extracellular matrix, 4% matrigel, and at the end of the differentiation, adding a little bit glucose to give them a more metabolites. And then we were able to very robustly generate these sophisticated structures at the end of the differentiation, which is around day 11, which contains two cavities, the cavity at the top and cavity at the bottom, and then have a cell dense area we believe is where the gastrulation happens, which I'm going to walk you through in more detail in the next few slides. So here just show just overall measurement of the different sizes of these type of structures. As you can see from day 1 to day 8, so the size is relatively uniform. But when we reach to day 10 to day 11, we see a variation of the sizes, so suggesting that towards the later time points, the efficiency, we see a lot of heterogeneity in terms of structure morphology, but the efficiency is very robust. We consistently observe about 60 to 80, even sometimes 80% of the efficiency starting with EPS cells. So very robustly and very efficiently, we can generate this type of structure. So here, just the image that we took on the day 8. So we took a scanning electron microscopy image. We remove the bottom part of this structure and just expose the interior. And just by looking at what people publish on, deposit online for human. So we're able to see some resemblance of the perigastroid are two, Carnegie stage 7 human embryo. Although they're not exactly the same, and also from this image, we cannot really tell anything, like just except for there is some resemblance, but nonetheless, it's very interesting to me. And by day 11, as I mentioned, we can really robustly and generate a lot of this type of structures. This is coming from one experiment. We can generate, we counted about 395 these type of structures looking very similar to each other, at this state. So it's a very robust system. So in the next couple of slides, I'm going to walk you through the different steps using this reporter line, the SOX2 citrine and SOX17 TUD tomato. Basically, SOX2 is green color, SOX17 is red color here. And starting from day 1, essentially all the cells uniformly express SOX2 because they're pluripotent. It's supposed to express this pluripotency-related transcription factor. And the day 2, once we expose these cells to differentiation media, some of the cells will become SOX17 positive cells. And then, as you remember, we remove one of the factors of the chiron, and then the cells starting to sort into different compartments. So day 3, you see they're sorting into 2 different compartments, the SOX2 positive cells, SOX17 positive cells, and by day 4, the sorting has been completed. As here, just took an image showing that they are uniformly in each well. You see these 2 compartments. One compartment is SOX2 positive cells, one compartment is SOX17 positive cells. By day 5, we're starting to see some interesting structures, organizations of the structure within the structures. So in many cases, we see this kind of arrangement. So in the beginning, we saw that this is SOX17 positive cells surrounding SOX2 positive cells, but it actually is not the case. It's because there's cavity formed in both compartments, and the cavity at the bottom, which is the SOX17 positive cells, is a little bit bigger, so that you can imagine it's like a boat sitting, floating on top of the like a liquid. And then that's why you see always, most of the cases, SOX2 positive cells are at the top, and SOX17 positive cells at the bottom. So they are not, this is a top view. If you look from the top, you give the impression that SOX17 positive cells are in the outer layer, but it's not the case. So in the side view, we can see that there are two separate compartments. And we also, perform immunostaining, look at the SOX2, SOX17. Again, they both express, the SOX2 expresses at the top, SOX17 expresses at the bottom. But interestingly, we're starting to see CDX2 positive cells. CDX2 is a marker typically used for trophoblast, but also the they are expressed in the amnion epithelium. So we believe, based on this and other evidence, that amnion cavity starting to be organized at this stage. We see the amnion epithelium marked by CDX2 positive cells, and then the epiblast compartment marked by SOX2 positive cells. And then you see this amnion bilaminar disc-like structures, which is marked by SOX2 at the top and SOX17 at the bottom. And here just this stack video showing that going through the structure, you can see clearly two different cavities. One at the top, amnion-like cavity, and at the bottom is surrounded by SOX17 positive cells, which is this yolk sac-like cavities. And in date 6 structures, we also observed primary germ cells being specified. And remember, we didn't add any factors at this stage, so PGCs arise on their own. So we use a reporter line as PRDM1, T52C. So whenever you see a yellow cells, they are primary germ cell-like cells. So the PRDM1 also expressed in the hypoblast cells. So whenever you see the red cells, they are the hypoblast cells. And then the T52C also expressing the amnion. So the green cells at the top is amnion. So from this image, it gives the impression that these PGCs are specified at the amnion and they are migrating towards the hypoblast. If this is true, this is very similar to what people describe in the monkey embryos. But again, we're not 100% sure, but we did capture these cells can migrate. Although in this video, it's really difficult to see where they migrate to, but we do see the yellow cells, which is the PGC-like cells, they're starting to extending polypodia and starting to migration. And by day 8, we're starting to see a more complex organization in the structure. We see, this 3-layered structures. Same gives the impressions, the SOX2 positive cells, and then SOX17 positive cells outside, but also we see SOX17 positive cells at the middle too. So we in the beginning we saw that maybe this is potentially the different endoderm cells that are out during the gastration process. We went on to perform some immunostaining analysis. First, we observed that indeed the anterior-posterior axis has been established. So using the SIR1 as one of the anterior marker, we observed that SIR1 localized to one pole of this structure. And then at the opposite side, we see T positive cells starting to popping up. So suggesting this anterior-posterior axis has been established. And also SOX2 are expressed for the anterior region, anterior ectoderm, epiblast region. And in some of the structures, we can appreciate from this image is that the T positive cells starting to elongate and lining at the center of this SOC2 positive cells. And then if you look at from a different angle, that give the impression that the T positive cells, maybe they're going downwards and through the process of delamination and migration. But again, we're very limited in terms of imaging these types of structures, getting really big. And so we are trying to get better structures to demonstrate that. And also from a different angle, we can see a trilaminar disc-like structure starting to form, which composed of the epiblast cells are yellow, and the purple, which is the T positive cells, and then the magenta is cyanide is at the bottom is SOX17 positive cells. So they give the impression of trilaminar disc-like structures starting to appear in this aggregate. And more interestingly, towards the end of the differentiation, about day 10 to day 11, we started to see a more complex self-organization. At the anterior side, we see SOX2 positive cells become this neural groove or neural plate-like structures. And at the posterior, we see a tailbot-like structures forming, basically have cells that are staining positive for T and SOX2. So these T and SOX2 double positive cells are oftentimes being recognized as a neuromesoderm progenitor AMP population, which is going to give rise to somites later on. So suggesting that in these advanced perigastroid structures, early organogenesis starting to be initiated. So we performed a single cell and seq analysis looking at the end time points, the day 11 perigastroid, we are able to see the major pyrogastulation cell types, including epiblast cells, including the ectoderm, and
कुछ नहीं, कल भी नहीं किया सर, 2-3 बार फोन भी
and
किए। हाँ, तो उसके
actually
मान लेता हूँ।
endodermal cells and some of the ectoderm cells as well. So, and more importantly, when we compare the integrated analysis of our human pyrogastaloid single cell data set with a published data set on monkey and human embryo, we see there's a lot of low overlap, suggesting that these indeed, the cell type indeed very similar to what people report in the embryos. So with that, I want to conclude this part. So by having a different cell types, starting cell type, extended pluripotent stem cells, and grow them in 3D and subject them to 4 steps of differentiation, we were able to generate structure, very advanced structure that can model this pyrogastulation development in humans. And so one thing I would like to point out is that these structures are not complete human embryo-like structure because it lacking the placenta cell types. So only 2 cell types to start with, the epiblast cells and the hypoblast cells. And we don't have the trophoblast cells. Unlike some other models people publish, especially from Jakob Penna and also the Magdalena's and Nick Good's lab, we have all 3 cell types composing their structure. But the issue with that is the efficiency is very it's not very high, it's about 2%, but in our case, we found our efficiency just using these 2 cells is very high, 80%, or in after further optimization is even more. So there is advantage and disadvantage of our model and versus the other model. So now I want to switch gear, and hopefully I have some time to talk about just the one other major direction in my lab, which is interspecies chimeras. So when I touch upon this topic, they're always generating 2 different reactions. One is excitement. Some students and postdocs are very excited about this very interesting, potentially animal or features that we generated in the chimeras. And the other group of people are very afraid. So they worry that we're able to, we're going to generate monster-looking like structure, animals like this with a lion head, goat body, and serpentine tail. But in the lab, though, interspecies chimeras doesn't look like a monster. It doesn't have superpower either. So it's normal-looking animals. So here, in this image, I take from here Nakauchi's image, and so we have 4 animals. On the left is a mouse, on the right is a rat, and in between are interspecies chimeras. So this guy here are generated by using red pluripotent stem cells inject into the mouse blastocyst and transfer into the mouse surrogate. And this guy here was done the other way around, using mouse pluripotent stem cells inject into the red blastocyst and generate, mouse red chimeras. Just by looking at this two, you can't help but notice that they're very different. So first of all, the body size is very different. So although both of them contain rat and mouse cells, but this guy here, the body size is essentially the same as the mouse, but this chimera is very similar to the rat. So suggesting this interspecies chimera can potentially help us to understand body and organ size control. And the other interesting aspect of interspecies chimera is developmental time. So a few years ago, we were able to generate the first horse embryonic stem cells from the horse blastocyst. We label the horse cells with GIP and then inject into the mouse embryos. So we were actually very surprised to find that some of the horse cells can robustly contribute to early mouse development. So this is the embryonic day 9.5 mouse embryo that contain a lot of green cells, horse cells. So this is without immunostaining, just the fluorescence. And we were very surprised by this because, horse gestation is very long. It's 11 to 12 months. And the mouse gestation is only 3 weeks. How is that possible? So the only one explanation is that some of the horse cells has find a way to adapt to the developmental time of the mouse. So suggesting there is a non-cell autonomous control on developmental timing. And potentially, interspecies chimeras can help us to understand what controls the developmental time. Maybe by understanding the mechanism, they can help us to differentiate proponent stem cell in a more accelerated rates in vitro, right? And the other very interesting aspect of interspecies camera is this. So the rat doesn't have a gallbladder, and the mouse has a gallbladder. So when you inject the rat proponent stem cells in the mouse embryo, will rat cells contribute to the gallbladder development? So the answer is actually yes. So these are rat proponent stem cell generated gallbladder in mice. So this suggests that the inability of the rat cells to generate gallbladder in their own development, during their own development process, is not because the rat cell cannot do it. It's just because the environment, the developmental niche, is not permissive for the rat cells to generate gallbladder. But in the species that have permissive gallbladder development niche, these cells from the donor cells can indeed generate gallbladder tissue. But more interesting, or more importantly, though, is this concept of growing human cells, growing human organs and tissues in an animal species, eventually can potentially solve the worldwide shortage of organ donors. Oftentimes, people want to use pig as a host because the pig organs are very similar to human in terms of size, physiology, and also in parallel, they're ongoing work with the xenotransplantation, try to use a genetically modified pig for transplantation purposes. But the problem is the chimeric contribution of the human cells in the evolutionary more distant host is very limited. Unlike the rat and cells in mice, you can see robust rat cell contribution to a variety of different tissues in this 18.5 months fetus. But in the case of human, though, we don't see any contribution. So suggesting that there are barriers to prevent human cells from contributing efficiently to a species, host species that are evolutionarily more distant from humans. So my lab, one of the main directions of the lab to study these barriers. What are those barriers? Can we overcome those barriers? What are the mechanisms underlying these barriers? So when I start my lab, I ask my student and postdoc to come up with the barriers that you can think about. So this is the list that they came up with. So there are potential cell size barriers, there are potential cell competition, cell adhesion, different developmental tempo, different proliferation rate and cell cycle. Maybe there's some incompatibility in terms of endogenous retroviruses, cell extracellular matrix incompatibility, ligand receptor incompatibility. There are potentially many, many barriers that we don't know. But I think it's important to understand them one by one. So today, just with the few minutes that I left, I want to talk about one specific barrier, cell competition. So cell competition is not a new concept. It actually has been described for more than 4 decades. In 1976, the first study of cell competition is reporting in Drosophila. So in a mutant called minute mutant, if you have the mutant, this is a mutant in the one of the ribosome protein, so the fly can be generated, and the fly wing disc can be generated normally. But when you place these mutant cells together with the wild type cells, then the mutant cells got eliminated through this process of apoptosis. So there is some kind of competition between one cell with their neighbor cells. So there are winners and losers in this context. So the winner cell will outperform the losers and take over development in some cases. So we believe that when we inject human proponent stem cells in an animal species such as the mice or the pig, and the human cells are treated as unfit cells and being targeted for elimination during the cell competition. So this is just a hypothesis, right? But we need to show that actually there is a active cell competition going on. But to study cell competition in vivo is very difficult because chimeric contribution is oftentimes random, and we know that the chimeric contribution of the human cells in mice or pig is very low. So it's not very practical to study this in vivo. So we had to come up with this in vitro system to understand their interaction. So luckily, we were able to establish this interspecies proponent stem cell co-culture system. So there's several culture systems that under the same culture environment, you can culture proponent stem cell from different species separately. they can maintain their proliferatory growth, they can maintain their propotency in the same media, right? And that gives us opportunity to grow them together, label them differently. So the human cells label in this case with the GFP, and mouse cell with M cherry or TD tomato. And then we can distinguish these two cell types. Now we can grow them together. So this is the first thing that we observe, which is a little bit surprise to us, is that the mouse cells, the red cells here, starting to attack the human cells within a few days co-culture. The human cells, due to this mouse cells presence, many of them undergo apoptosis. This is very shocking to us because why these two cell type has to interact in such a way, right? They can just grow on their own, without bothering each other, but somehow there's this competitive interaction. So we counted the human cells from day 1, day 2, day 3, day 4, day 5, either in co-culture as a blue line here, or separate culture. The same number of human proponent stem cells are plated. But then one is in co-culture, one in separate culture. And starting from day 3, we start to see there's a difference. The human cell grow by themselves, they continue to expand, proliferate. But the human cells grow in the presence of mouse cells. The total cell number is greatly reduced. By day 5, essentially no human cells has been found in the co-culture. But in the case of the mouse cells, doesn't matter. In the presence or absence of human cells, there is no difference. They grow at a similar speed. And we want to know whether direct human and mouse cell contact is important for this competitive interaction. So we grow them together, but in this case, we separate them physically by growing human cells on top or at the bottom, mouse cells on top of it, to using this transwell co-culture system. And in this case, though, we didn't observe human cell apoptosis. So suggesting that they need direct contact with each other in order for this competition to occur. And also, we wanted to know whether secretive factors are involved in human cell death. We collect the conditioned media from human cells, or from co-cultures, or from separate culture mouse cells, and we use the conditioned media to grow human cells. And as you can see here, there is no difference. So suggesting growth factor is not at play here. There's something else. We don't know what it is. But if you block the apoptosis in human cells by either overexpressing BCL2 or knock out or knock down EP23, we can overcome the human cell death. So this is encouraging because we know now the human cells is dying through apoptosis. So this is one of the classical outcome of cell competition, is dying through apoptosis. But we want to know a little bit more what other mechanism at play. So this is what we did. We isolated the human cells, either cultured on their own or co-cultured with mouse cells, and did RNA-seq analysis and then compare the transcriptome. So one of the pathway that jump out immediately to us is this pathway called NF-kappa B pathway. So NF-kappa B pathway was quickly activated in the co-cultured human cells when compared to separate cultured human cells. And NF-kappa B pathway can be triggered by several different upstream signaling. One of them is through toll-like receptor, this innate immune response pathway, and through this adapter protein called MyD88. And indeed, when we perform the Western analysis, we see during co-culture, the I-kappa B alpha, which is inhibitor of the NF-kappa B, is downregulated, and phospho form of P65, which is a core component of NF-kappa B complex, is elevated, suggesting indeed this pathway is activated, right? Not just suggested by the RNA-seq analysis. And this is interesting because several years ago, Laura Johnston's group in Columbia University found in Drosophila. So in 2 different cell competition model, they all impinge and converge on this NF-kappa B pathway to regulate loser cell death. So suggesting maybe there is this ancient cell defense mechanism, innate immune response mechanism are at play here to eliminate the human cells. Now, by knocking out the P65 or MyD88, the core component of the NF-kappa B complex or upstream adapter, we can completely overcome the cell competition between human and mouse cells. And here, this video shows that by co-culture the mouse cells with P65 knockout human iPS cells, human cells can now coexist with mouse cells. There is not much apoptosis going on. So we greatly improve the human cell survival in the presence of mouse cells. But by looking at the video, you can't help but notice that there are another barriers at play here is that all the human cells are growing together, most of them, right? Mouse cells are growing separate. So suggesting the cell-cell adhesion are another barrier that we need to overcome. But I don't have time to talk about today, which is currently ongoing in my lab to overcome cell competition barrier. But the key question is, by overcoming this, F-coupled pathway, whether we can improve the survival of human cells in the mouse embryo, right? All I talk about is in vitro. So indeed, we were able to demonstrate that. So MyoDDA knockout or P65 knockout human iPS cells can survive much better inside the mouse embryo, either before implantation or after implantation. They can even differentiate into early, in this case, ectodermal progenitors labeled by PAC6. So that's just between human and mouse. What about other species? So luckily for us is that we have, under the same culture condition, we can grow monkey, mouse, rat, human, and cow proponent stem cells, the exact culture condition. So we found that in all the co-cultures between rodent and primate, all the time, the rodent cells are the winner cells. And the monkey or human are the loser cells. But when you grow proponent stem cells from closely related species like a rat mouse together, there is no competition, suggesting this competition occurs between evolutionarily more distant species. But more interestingly is the cow. So when we grow the cow cells together with mouse or rat cells, expectedly, cow cells are the loser cells. Then we outcompete by the rodent cells. But when we grow human and cow cells together, now interesting aspect is that human cells now switch role from the loser to the winner. So suggesting that it really depends on who they co-culture with. So humans can become a loser or the winner, depends on the co-culture partner. So with that, I like to summarize this cell competition study. So by developing this interspecies proponent stem cell co-culture system, we discovered a really unexpected cell-cell competition between species that are evolutionarily more distant from each other. And this cell competition doesn't really occur between species that are close related together. I didn't have time to go into the detail, but we found that the cell competition only occurs in the prime proponency, but not the naive, but not after immediate differentiation. So it's very specific, this peri-graduation development of the epiprocess. So with that, I'd like to thank all the people in my lab. Without them, I'd not be able to share with you this very interesting story today, and also all the funding sources that got us here. And hope I have some additional time to answer any questions that you may have.
Jun, it's a very interesting talk, covering 3 different. Do you hear me?
Thank you. Yes.
Yes.
Sorry, I take a little bit too long.
It's okay. It's okay. So we actually got the opportunity to hear about the human blastoid cells, about the perigastruloid co-culture conditions, as well as the interspecies chimera formation. I mean, if any participant has got any questions, yeah, you give it. Maybe 1 or 2 questions we'll take up.
Sure.
John, one moment. Prashant is a student from my lab, and
Okay.
he has got opportunity to hear you before about and he read your work fully about the interspecies, and he was the one who actually proposed your name to come on the workshop.
Well, great. Thank you. Yeah.
Prashant, please.
Dear Dr.Jun Wu, I'm a big fan of your interspecies chimerism work you did in Salk Institute, and I have 3 questions. So you did gene knockout for the organ development in pig embryo. You injected the human iPSCs. So the human organs will grow inside, but the vascular system will be contributed by the pig cells. So how to deal with this? And another thing is...
One by one. Okay.
Yeah.
Question.
Yeah. So that's a great question. So we don't know the answer at this stage, but I can I can speculate what potential solution is. So in the future, so at this stage, even though in medicine study, people claim that it's generate the organ, but it's not a full organ, right? I mean, when I talk about it's between mice and rat, but not humans, not there yet. But even between mice and rat, it's not a full organ. It's just the parenchymal cells or the cells that are you empty the host niche for, right? The endothelial cells are still there from the host. So endothelial cell, as you know, is a big problem for immune rejection. But I think one potential solution is that you can combine this process competition approach together with the xenotransplantation approach. So we can potentially, in the future, using the pigs that are already genetically modified to reduce the immune barrier, and then use those pig as a host to enrich more, human cells in that organ. We're not going to get 100% human organ. We're going to get maybe 80% or 90%, depend, on the tissue, depends on organ. But
And another question is, after injecting the human iPSCs, the cells will migrate, the pattern of migration will vary. In your rat and mice model, there was a contribution to brain and multiple organs like liver and kidney. In your pig model, there was no contribution to the brain cells. So why there is the variation is there? So you reported that it will not
yeah,
migrate into the brain.
so in that we didn't observe, first of all, the contribution is very low. I think it's in order to identify the positive human cells that they really need to do a lot of immunostaining, a lot of assays to be to demonstrate that. It may be possible that because we did analyze enough embryos, and at least from the embryos that we observed, we didn't see the contribution to the central nervous system. That I cannot exclude the possibility that in the future, if we increase the human cell chimeric contribution, that maybe some cells can be potentially contributed to the central nervous system. That obviously that will raise some ethical concerns, right? I think it's equally important to develop a strategy, try to prevent the human cells from contributing to those tissues that are more sensitive, ethically more sensitive. We're developing some genetic means, for example, knockout genes that are prevent the human cells from contributing to the ectoderm, or kind of help guide the human cells to bias the human cells towards only become mesoendoderm by briefly treating them with certain chemicals to change their differentiation trajectories initially. But again, we don't have a good answer at this stage. Yeah.
Prashant, we will restrict your questions and you can contact him by email. Okay. I have one
Thank
quick question for you.
you.
When we talk about the competition between two species cells, we talk about so how about the pig and human cells? Who
Yeah.
winner is?
So pig human cells, I think one of the issue is the pig ESL cannot be cultured in the media that we use them for competition. So we were not able to do that experiment. But there are some other culture condition that can co-culture human pig cells. But those culture are more like a naive state or more like an earlier state. We didn't observe competition, which is not we should kind of expect it. But we do know that if we inject the human cell into the pig embryo, they are eliminated. Yeah, there is no human, not much human cells can survive after the gastulation. I think that still suggests there's maybe competition, maybe some other form of elimination of the human cells, because during pig development, the trophoblast, the embryo starting to elongate,
Okay.
and that is very different from human embryos. So that may posing some of the challenges for the human cell to survive as well. So we again we don't know the answer. But we do know in vivo that human cell can be eliminated by the pig embryos.
Okay, wonderful. Thank you so much, Dr. Wu. And hopefully
You're
we'll
welcome.
be again meeting on such a platform, maybe listening to your new work and new things. And if we do not have any other questions at this moment, we'll close the session over here.
Dr. Dhara, can I have one question, two questions?
Okay, yes, please. My colleague from Bangalore, Dr. Ghosh,
Sure.
is here. Okay, please.
So I have basically two questions. One of your slides, you said that you have cultured the horse ESLs. How they are different in terms of media composition than your mouse or human ESL culture system? That is the first question. And second question, in another slide, you have shown that the amniotic cells are migratory. So it is similar to your PGC. So it is strike to me because of the reason we always know that PGC is originated from the yolk sac cells. So does this finding contradict?
Yeah, so a very, very good question. So the first one, if I understand you correctly, so you're asking the cow cells growing in the media
Yes.
with the human, what are the characteristics, right? Are they similar or not? So at least I can tell you is that they're all prime cells. So they express all the classical prime potency markers. But obviously, there are species difference between even in the same culture conditions. There are some species difference. The morphology of the colony is very different from human mast cells, and they have this spreading more loosely contact colonies. And like human, they are more forming a colony, right? And the mouse cells are actually they forming a more like more compact colonies than human. So morphologically, they're different. But some of the molecular marks are similar, I have to say. And for your second question, very good question. And we really don't have an answer for it right now because in non-human primates, some people reported that the PGC can be specified from amnion epithelium. But in human, because we don't have human embryos to do that work, we really don't know what's going on. So the belief, I believe is that the yolk sac. So first, you brought up the yolk sac. So actually, the yolk sac is true that after specification, they might migrate to the yolk sac, to the hypoblast region, right? To hypoblast region. So that's our model suggests. But again, we don't know for sure whether this is the case or not. So in Jianping Fu's model, when he used the amnion sac model, he observed that the PGC can be specified from both the amnion epithelium and the epiblast. Again, we still don't know whether this is true or not, because all these in vitro systems can be, some can be artifacts, some can be, cyto using the prime human pluripotent stem cells, induce them into intermediate mesoderm-like cells, right? And then use those cells to generate PGC-like cells. So unfortunately, I don't have a good answer for your question, but I think that's one of the most important, I think, the question that people want to ask is that where's the origin of the human PGC? Where do they specify, right? So that I think with more sophisticated embryo model, we potentially can answer the question, but yeah.
Yeah, thank you very much, Dr. Wu. And I also would like to thank all my colleagues who have joined online from different parts of India. They have joined. Some of them have about more than 100 people registered to attend your talk, Dr. Wu. About
Thank you.
110 people have registered for your talk. We have actually put up a system that we wanted to, but maybe because of too early in the morning, maybe they are not able to join or for some reason, whatever. Also, you went on live in the YouTube channel at this point when your entire talk. So I once again thank you for sparing your time to give this to these participants, and maybe some of them will contact you for their personal questions and queries on your talk as well as other academic interest.
Yeah, absolutely. If you have any question, feel free to contact me. Yeah, I'd be happy to address any of them. Yeah.
Thank you so much again.
Yeah.
Okay, with this we'll
Bye-bye.
close. Bye. Good to see you again. Okay.
Good to see you again. Yeah. Have a good day. Yeah.